Hey everybody, Mr. J here. Today we are in week 7 review. This first one will be over the adaptive immune system, specifically our lymphocytes. So we're going to be talking about our B cell activation, as well as how our T helper cells or CD4s are going to help kind of spur the B cells on to immunity. Secondly, we're going to talk about antibodies. We're going to talk about their structures, as well as what their structures kind of do for their functions. And then finally, we're going to talk a little bit about passive versus active immunity, as well as the primary and secondary response based upon when you get an infection and if you make antibodies for it. So let's jump right in. Hopefully this is helpful. So first off, whenever you have any sort of infection, if you have a bacteria in your system, if you have a virus in your system, those things will have what's called antigens on the outside of the cell. So it could look like a little bacteria that has these proteins or glycoproteins kind of hanging out on their cell membranes. And these antigens to your body are considered foreign. They are unnatural. We don't like them. So a lot of the times, these bacteria will get destroyed, targeted for destruction. But let's say you have a big infection. So now a lot of bacteria are getting destroyed. You're starting to get an inflammatory response. And a lot of their free antigens, after being destroyed, will be floating around in your system. We also talked about how they can be used as antigen-presenting cells, like dendritic cells will do this, where they'll actually take some of the antigen from the bacteria and present it to different lymphocytes. This isn't the process we're talking about today. What we're going to talk about is how the B cell itself is going to act as its own professional antigen-presenting cell. So what the B cell will do is it will take in this antigen from the bacteria or whatever infection it is, and through its endoplasmic reticulum will join it with something called MHC2. I like to think of MHC, the major histocompatibility complex, as a big flag holder. Okay, that flag holder is going to hold the flag, which will be the antigen. So this flag holder, MHC2, so if I can write that in really small, MHC2, is going to place its flag, the antigen from the bacteria, into its flagpole. Okay, great. So now we can consider this B cell a professional antigen-presenting cell because it's presenting an antigen. Who's it going to present to? Well, it's going to present it to the T helper cell. So if we flip this picture, the B cell is going to find its way to a T helper cell, also called a CD4 cell. And I'll get to that in a second. So let's say this B cell comes into contact with the T helper cell. So it got that MHC2 on its outside. It's going to present that antigen to the T helper cell. And so these two things are going to bind together nicely. So both the B cell and the T helper cell. Once the T helper cell connects with the antigen and the MHC2 complex that the B cell is presenting, it is going to stimulate the release of what's called interleukins. So interleukins, all of these little molecules are going to be basically secreted out of this T helper cell, and it's going to go to the B cell. This will stimulate the B cell to differentiate. And we'll get to that in a second, but what I wanted to connect is that why it's called a CD4 cell is because this complex right here on the T helper cell, this is considered the CD4 protein complex. Okay, there's several different chains of proteins. I want to say carbohydrates as well, but we're just going to say it's CD4 just to simplify. Okay, so once these guys bind, another thing that I forgot is this T helper cell has to have bound to this antigen before. It has to have seen it before. So basically it's like, oh, I see this issue, and now I see my friend B cell also has an issue. They combine together and they start freaking out. So this is when the freak out starts. The T helper cell releases interleukins. That will stimulate the B cell to differentiate into two different types of B cells. Most of the time, it'll turn into, so 75% of the time or so, it will turn into plasma B cells. These plasma B cells will be the antibody producers, and we'll get to antibodies in a second. So these are going to be producing a massive amount of antibodies, which will bind to the antigen, basically targeting things for destruction, covering them up, preventing the infection to bother us anymore. Whereas 25% will turn into, well, that's a terrible 5. 25% will turn into what's called memory B cells. Okay? So memory assumes that they can remember something. That makes sense. And memory B cells will remember the antigen. And this is beneficial when we have a secondary infection. So say you get infected by the same thing twice, the secondary response will be elicited by the memory B cells. They'll basically recognize that antigen and be like, we've seen you before, let's start making antibodies for you. So this will stimulate a faster secondary response. Okay. So basically the front line that's actually fighting the infection off will be those plasma B cells. They'll be producing antibodies, and we'll get to, I put antibody producers. And these antibodies are going to be the direct link between destroying our infection, our bacteria, viruses, whatever it is, neutralizing it, and what have you. So these are the frontliners, and then these are going to be kind of special ops. They're going to go back to the fort, and they're going to just wait for this infection to happen again, and they'll be ready and primed to do it. So let's get into that a little bit. When we make our B cells and we start producing antibodies for a novel infection, a new infection, this is going to be called the primary response. This is the first response to a new infection. This primary response, so basically the time it takes from the time you get infected to the time you're producing antibodies for that specific pathogen, takes anywhere from 3 to 14 days. Okay, it takes 3 to 14 days, so a little bit of time, right? Well, the secondary response, so say you get the infection again like a year later, right? You're going to have some memory B cells chilling out, waiting for that same antigen to be presented. If you get infected by that same virus or bacteria, this is now going to be considered the secondary response. And this takes hours to about 2 days. So basically, you are cutting down the speed of response so that you start producing antibodies much faster in response to that same infection the second time as you did the first time, okay? And that helps you fight off the infection very, very quickly. Okay, so that kind of goes into what I want to talk about with passive and active immunity. So we're going to go here first, and then I'll talk about why antibodies are so important for fighting off infections. So when you get a vaccine, right? When you get a vaccine, you are basically getting a weakened part, let's just say, of a virus. Okay, a weakened part of a virus. The virus isn't going to bother you, but its antigens will get into your system and elicit this immune response. So a vaccine is kind of unnatural. It's an artificially made thing, right? But your body is going to be actively producing the antibodies to fight it off. So this would be considered a type of active artificial immunity, which would be vaccines. Again, you are still producing the antibodies, which is the definition of active. You produce the antibodies. But it came from an outside source, right? We made the vaccine, we put it into our bodies so that we can make antibodies and then fight off that infection. Natural active immunity, you could probably guess. This is just when you get your normal infection and you will make antibodies for the pathogen and you will fight it off. So that would be a type of natural active immunity. You may be asking, okay, what's passive then? Well, if active was you are producing the antibodies, passive means you get the antibodies from another source. Okay, so here you are getting antibodies from another source. Let's think about this. When you're a baby and you're nursing, so you're actively drinking breast milk from mother, you are actually receiving some of mother's antibodies. That's a good thing because you don't really have an immune system. You haven't been primed. You haven't gotten an infection in your first 3 months of life, right? But you're going to, and you need to be able to fight it off when you are ready. So you can get direct antibodies from breast milk. Kind of cool. And I would consider that to be a pretty natural process, right? Breastfeeding for an infant. So breastfeeding, and you get antibodies from mom. So I'll say get ABs from your mom. And that's a very beneficial process. But we can also directly infuse your body with antibodies. You may have heard of, I think it's monoclonal antibody treatment for people who have COVID-19 and they want to fight it off a little better. Maybe their immune systems are pretty weak. So we can take antibodies from somebody else, okay, take them out of their body, put them into somebody who needs them really badly because their immune system's not fighting it off very well, and then those antibodies are going to do the work for them. So this is a type of passive artificial. It's not natural, and it's passive because we're getting antibodies from another source. So antibody infusion from another person. So that's the differences between the types of immunity. Lastly, antibodies. Okay, what's the big deal with antibodies? I'll tell you what. Antibodies are going to bind to these antigens. So what these will literally look like, if I have my antibody coming into these antigens, is they will bind perfectly to the outside of these antigens. So let's say it looks like this. And just the fact that our B cells can literally produce antibodies to fit perfectly down to the atom on those antigens is absolutely fascinating to me. Absolutely fascinating. The fact that they know how to mechanically make these little antibodies to fit right in there. And when they do that, they are going to do one of several things. They can neutralize the antibody. They can opsonize it. So this is basically targeting it for eating from macrophages. And they can also elicit inflammatory responses so that we make more and more and more and more antibodies in response to infection. And it can also just target it in general for destruction from other cells. Okay? How does it do it? Well, I said it has to bind to the antigens. So if we look at a structure of a general antibody, and there's 5 classes, I'm just going to go through a very general one. The 5 classes will deal with different types of infections. So you can study that if you ever take an immunology course. But antibodies will have this in common. They will have what's called a variable region. These are kind of like the pegs that are going to go into the antigen. And they're variable because they're going to look differently depending on which antigen they are binding to. Just like I drew this one kind of like a weird shape that fits into that antigen nicely, that will be a difference within the variable region. However, you also need to have constant regions to basically tell macrophages to eat this thing, to tell basophils to stimulate this inflammatory response. So we need to have some sites where those guys can actually bind. Those are found here. Okay? So in these binding sections, and I forgot one, I'll tell you about it. This binding section is a binding site for macrophages, so the big eaters, the ones that will actually eat the infection, as well as basophils. I think of baso as burning because they are the big proponent of inflammation. So let's say an antibody pops onto an antigen, pops onto this bacteria. Well, a basophil can come along and bind to this site and basically say, oh shoot, there's an infection. Antibody just bound to this thing. Let's ramp up the inflammation response. Okay, so basophils start making histamines and all these other chemicals that basically tell the body, hey, we're under attack, we need to fight this off. Furthermore, it can also be a docking site for macrophages. So say it pops there, and then the macrophage binds to the antibody here and is like, oh shoot, this just got tagged because it's foreign, I need to eat it. And the macrophage will eat it. There's also a binding site, I'll put it on this side so I can write, and this is a binding site for the complement proteins. I didn't talk about that in this lecture, but the complement cascade is basically several proteins circulating in your bloodstream that can, I would say non-specifically, but also non-specifically bind to foreign things and trigger inflammation and trigger the killing of these foreign substances. So they're just proteins floating around, and this is a part where the complement protein can bind here and basically stimulate a further cascade to fight off the infection. All right? So this is a very general run-of-the-mill how we fight off infections. I hope this was helpful. Please like and subscribe, comment for any questions, and I appreciate you watching.